and welcome to online lecture 2B here in your Biology 112 semester. Today we'll be in the Chapter 27 PowerPoint, Chapter 27, looking at slides 26, perhaps as far as 46, but we may go a little short of that, but at least 26 through 40 or so, guys. Our topics today include mucilage, gram staining, bacterial and archaean motility, bacterial and archaean reproduction, and we might get into a little bit of nutrition and perhaps metabolism towards the end. But let's start here on slide 26. Just kind of go back a little bit here, and then we'll move forward into the rest of the slideshow. So let's go ahead and see if we can get this thing to start going here. So we got this slide entitled Mucilage. Yeah, just doesn't sound really good, does it, guys? Well, yeah, the name kind of describes it pretty well. Mucilage is very similar to mucus. Why? Because it's composed of polysaccharides and protein. A lot of times it's a combination of both. It varies a lot in consistency. There are times it's kind of like mucus, but there are times it's more like a gel or like a gel capsule. So it can be fairly rigid. It really varies quite a bit. And if it's thick and almost capsule-like on the outside of the bacterium or archaean, but typically bacteria, it's called a glycocalyx. So glycocalyx, mucilage, capsule, we're talking about the same things here. Now the mucilage is actually secreted from the bacterial cells. It's not actually directly a part of the cell. This is a cell product. Now the big question is, why do bacteria produce this mucilage, this mucus-like substance? Well, you can imagine it's used to evade host defenses. So the thicker the glycocalyx, the more resistant the bacteria is to your immune system's attack. In fact, you can often tell which bacterial species are harmful just by looking at the thickness of the glycocalyx. If it's thicker, much more likely to cause disease. If it's thin, your immune system typically can handle that in many cases. And what else? Well, also, mucilage holds the colony together. And so by holding the colony together, this allows the colony to exchange in cell communication. It can help aquatic species to float like as a mat all together. It can bind mineral nutrients and, of course, repel attacks. So there's a lot of different things that mucilage can do. And when the mucilage is released, the colony is glued together, and it can help to form what are called biofilms. A biofilm is simply an aggregation of microorganisms that secrete adhesive mucilage, basically allowing them to glue themselves or stick to surfaces. So dental plaque is probably the most famous biofilm. We're constantly having to remove that biofilm twice or once a day, however often you brush. You're removing that biofilm. That can be a very harmful biofilm, because when that biofilm forms on your teeth, that traps the bacteria up against your teeth, and they break down sugars that you eat, and they convert those sugars into acid, and the acids wear away at tooth enamel. You can also get biofilms in large industrial pipes. So in pipes, the biofilms start to form up on the end of the pipe, and they develop and grow along the inside of the pipe. They start to break down and corrode the pipe. So eventually you wind up with cracks in the pipe and leakage. So we've had some pretty bad problems with biofilms developing in industrial pipelines. But what are they good for? Why a biofilm might be good? Is there anything good? Well, yeah, they help to stabilize soil surfaces. They help stabilize the enriched sand, where the sand may have no nutrients at all. Having a biofilm there helps give that sand a little bit of a organic base and makes it a little bit more nutritious and more livable. And they also help in the formation of mineral deposits. So they help minerals that are floating around in the water to become deposited in a certain location, which enriches the nutrients in the local environment. And again, you can imagine how they might stabilize soils. These layers biofilms forming on the surfaces of soil prevent soil erosion in many cases. So that's mucilage, and again, very important in the microbial world. This, folks, is a biofilm. So you can see in this picture here, you've got the bacterial cells. That would be like these red cells in green, these bacteria, and there's also bacteria, smaller bacterial species in blue. We have at least 1, 2, 3 species of bacteria, perhaps even more. And so we have the bacteria growing in high concentrations in this large colony, and these spaces here would be where we have our carbohydrate protein mix of the mucilage. So the mucilage would fill in the spaces, kind of holds everything together. Yeah, just this picture right here will motivate you to brush your teeth, won't it? So if you struggle with dental hygiene, just remember this picture, guys. You'll be motivated for much more than you probably have been. Let's go to slide 28. So here on slide 28, we're changing the topic a little bit, and we're looking now at structures of the cell wall in archaeans and bacteria. Now, we can say that most our prokaryotic cells, including archaeans and bacteria, have a rigid cell wall outside the plasma membrane. So if this is the plasma membrane would be in the cell wall, and then, as I mentioned before, your would be capsule, or your glycocalyx, or your mucilage. So again, first ring would be your plasma membrane, the second would be your cell wall, and then that very outer layer here would be your glycocalyx capsule, your mucilage, you might call it there, right? So there are many layers in the outside of bacterial cells. Anybody know why bacterial cells have so many layers out here? Well, if you think about it, guys, you know, they're unicellular. So what is just outside the cell? Yeah, the extracellular environment. So when you have the extracellular environment in direct contact with the cell, you need to have a lot of layers to protect you, whereas our cells don't have nearly as many layers. Does anybody know of these 3 layers, what do our cells have? That's right. We only have the plasma membrane. We don't have cell walls or a glycocalyx capsule. So what's the purpose of the cell wall? Well, it helps maintain the cell shape and helps protect it against attack from that extracellular environment. And because it's rigid, it does, again, provide a consistent shape. Now, why would that be important? Well, if you maintain your shape, you can maintain your function, because structure is very, very influential on function, if you remember. So we want to maintain our cell shape so we can continue to function as we should. Also helps the cells avoid lysis in hypo solutions. What does that mean? You remember? Well, first, lysis means to pop. So if we have cell lysis, the cell ruptures and basically pops, sort of explodes. Now, why would a hypotonic solution cause that to happen? Does anybody know? What would be an example of a hypotonic solution? Can you example how distilled water, right? Something does not have many solids in it, like pure water, something very diluted. And so the water will rush into the cell because the cell is hypertonic compared to the hypotonic surroundings, and the cell will expand and then even pop. So the cell wall will stop that from happening. Our bacteria use protein, so instead of having a cell wall made of peptidoglycan, some archaea, archaeans, all of them, and some bacteria will then just use proteins. But most bacteria use peptidoglycan. Now, what is that? Peptidoglycan is a polymer, so it's a long chain of, sort of structure, and its macromolecule makeup there. And it's basically composed of carbohydrates that are cross-linked by protein, which makes them rather strong. Okay, so that's a little bit on the cell wall. Now we're going to compare 2 basic groups of bacterial species that have slightly different cellular wall structure. Okay, so let's go on to the next slide here, Gram stains. Okay, so the cell wall structure will determine whether a bacterium is gram positive or gram negative. let's first talk about gram-positive bacteria. Gram-positive bacteria, when they are stained, the gram stain technique, become purple. So if you see purple bacteria in a slide that's been treated with gram stain, you can bet it's a gram-positive bacterial species. Why does that occur? Well, it turns out that if you're gram-positive, you have a very thick peptidoglycan layer. You have that very thick carbohydrate polypeptide cross-link structure. So it's quite thick and very resistant to immune system attacks. So some of these guys can cause disease as a result. And having that thick peptidoglycan layer absorbs the purple color, which is caused by a special indicator stain called crystal violet. So crystal violet, purple violet color. And crystal violet, when in the absorbed by the thick peptidoglycan layer, will remain purple. And so these cells are stained a dark purple. They are very vulnerable to penicillin. Penicillin is an antibiotic, as you know, that's very good at interfering with the production of the peptidoglycan. So penicillin will shut down peptidoglycan production, and since these bacterial species have such thick peptidoglycan layers, if you lose that because of a lack of peptidoglycan production, you will then not be able to evade the immune system of the person and you'll die. So penicillin is very good at treating gram-positive bacterial species. What about gram-negatives, including things like proteobacteria? You know, we talk about proteobacteria. A lot of organisms cause disease, right? Like stomach ulcers and cholera and salmonella. These are your proteobacteria, all gram-negative. Again, just to remind you, gram-positives would be things like the firmicutes and the actinobacteria. And also you'll find among the gram-negative, you'll also find cyanobacteria. So there's some of the groups we've talked about before already. So gram-negatives, well, they have a peptidoglycan, so they have a thinner peptidoglycan layer, and they have a thin outer envelope of lipopolysaccharides. So you might think, well, these guys are more vulnerable maybe, like these bacterial species would be less dangerous because their peptidoglycan layer is not as thick. However, in addition to the peptidoglycan layer, which is thinner, they have a thin outer envelope of lipopolysaccharides, which helps them to resist penicillin. So you have to use other antibiotics to treat gram-negative bacteria. So you will not get penicillin to treat your stomach ulcer because your stomach ulcer will be caused by a proteobacterium, which is gram-negative. So in the gram-negatives, they tend to be pink. Why? Because the peptidoglycan layer is thinner, therefore the crystal violet doesn't get absorbed as well, and it washes out, it being more of a light color. So you can look at a glass slide and quickly tell, looking at the bacteria, whether they are gram-negative or gram-positive just by the color. Pink for gram-negative, purple for gram-positive. Okay, let's see if we can look at some pictures now of what this might look like. So, oh, looks like we just lost our slideshow for a second there. Let's see if we can back up. Okay, so here, guys, you can see gram-positive bacteria, right? So notice they are very pink color, whereas the gram-negatives are pink. And so this is probably E. coli over here because it's E. coli is also a gram-negative, and it's got the rod shape. So if you see a rod-shaped bacterial species that's pink, it's probably E. coli, which is living inside of you. Over here, this gram-positive, this could very well be Streptococcus because it's circle, sort of grape-shaped, and you could see the dark stain there showing it's gram-positive. So the gram stain is just a way to help us figure out which bacterial species we're dealing with, and can be used to diagnose disease. Like if you get strep throat, they can see based on the shape, the cocci, or the cluster of grape sort of spherical shape, plus the gram-positive dark stain, that purple color, will indicate that you have strep. So we use these things to diagnose all kinds of diseases. So let's go ahead and move on to the next slide, slide 31. This shows you even more pictures of what things look like. So this is gram-positive. Look at how thick the peptidoglycan layer is. So you have all this carbohydrate right here, and then you have these long skinny peptides making this whole thing stronger. And then below that, you have your plasma membrane. Whereas in a gram-negative, you have a thin peptidoglycan layer, right? This is not nearly as thick as this over here, right? So much thinner peptidoglycan layer, but what's the catch? You have an extra layer on top called the lipopolysaccharide outer envelope, and this makes things very strong. So if penicillin is trying to attack here, that lipopolysaccharide outer envelope will resist and cause the bacteria to be able to survive. You can imagine that if we're using a gram stain, this is going to stay more of a purple color. The crystal violet stain will become purple, whereas the crystal violet's not going to just stick. Plus the lipopolysaccharide envelope's in the way, so you want it with more pink color here. Okay, guys, let's continue on here. Moving on to slide 32. Let's talk about motility. How do these microbes get around? You might wonder. Well, there's some really neat different techniques that they use to get around. First of all, they have motility, which means the ability to favorable conditions, or the ability to move to different locations. The purpose of motility is to move to favorable locations, like places where we might find food, or to avoid things like toxins. These bacterial cells, archaean cells, need to get out of the way of toxins. They want to go toward food, or perhaps toward oxygen, or even away from oxygen. There's a lot of different reasons why they might need to get around. They respond to chemical signals. They actually respond to chemical gradients. So if we have a high concentration of a food item or a nutrient and a low concentration over here, bacteria can detect that, notice the gradients, and drawn toward high concentration of nutrient. How do they actually get around? Well, some swim, some twitch, some glide, or some just simply adjust their flotation. If you guys remember those gas vesicles, this allows bacteria to float to the top or sit down to the bottom, depending on how full the gas vesicles are filled with gases. So that is motility. Let's examine this a little closer, though, and let's look at some examples. That bacterial flagella, which I'm going to show you a video of in just a second. The flagella is one of the most common structures used to help bacteria swim in aquatic environments. So this is effective where we have water, in moist areas. So any bacterial species living in moisture, high levels of moisture, will likely or fairly likely have flagella or flagellum. Now, they're not the same as the eukaryotic flagella. They're not quite as sophisticated. Like, we do not have a plasma membrane surrounding the flagellum. We also do not have microtubules on the inside. If you recall, there's a oops, didn't mean to back up there, guys. Let's back it up here. There we go. You have the 9+2 arrangement. If you guys recall back from Biology 111, let me see if I can get our thing back to working here. Okay, looks like we got it fixed now. Okay, so anyway, we have the 9+2 arrangement of microtubules on the inside of the flagella and even cilia and eukaryotes. We do not have the 9+2 microtubule arrangement here inside of the bacterial flagellum. No dining arms either. So there's no plasma membrane, there's no microtubules, and we don't have the motor protein dynein on the inside. It's kind of like an outboard motor. The way it works here, guys, is hydrogen ions here flow across the actual motor. They go through channels with the motor. So the hydrogen channels are able here, and you can see there's lots of them. There's one there, one there, one there. Hydrogen channels all along the motor. These hydrogen ions were actually pumped outside of the cell before. And so hydrogen ions were forced through an electron transport chain, kind of like what we saw in photosynthesis and aerobic respiration. And so the hydrogen ions want to flow in And when they flow back in, going from high to low concentration through like an osmotic process or something like a diffusion, they flow through these channels. And as they flow through, this powers the motor. The motor then begins to provide the energy to the hook. Then the hook begins to rotate, and then the hook's rotation connects to the filament, which spins around very much like an outboard motor. So when the prokaryotic flagella is operating, it spins around like an outboard motor, whereas the eukaryotic flagella tend to just sway back and forth. It's quite a bit of a difference there. So let's look at an actual video of this. Let's go ahead and see if we can show you guys a short video clip of what the bacterial flagellum looks like, how it operates, and I think you'll appreciate it a lot more once you see this video. There's the motor, there's the hook, and there's the filament. And there's your filament. And here we go. Here's the action. Hydrogen ions flowing through the motor protein channels. And look at it spin. Filament spinning, hook causing the spin. Very neat. All right, quite a bit different too than the eukaryotic flagella. Very neat little video there. You could see very clearly how the prokaryotic flagella operates. But sometime we'll look at the eukaryotic flagella. You guys can see that in more detail as we get further into the semester here. And then, just while we're talking about the flagella, I mentioned the flow of hydrogen ions. That's what powers the bacterial flagella, but for archaeans, it's the hydrolysis of ATP. So ATP provides the power for the archaean prokaryotic flagella, so a little different there. All right, let's move on to slide 34. Here's some pictures of some flagella found in Vibrio. Looks like is it Vibrio? No, it's not Vibrio. It's the this particular organism here provides or causes, I should say, food poisoning. Some type of a yeah, it is Vibrio. I was right. It's a food poisoning, a seafood poisoning, and it only has a single short flagellum. Can you find it? Can you see the short flagellum here in Vibrio parahemolyticus? Parahemolyticus, and this is again causes of bad seafood poisoning. And then over here we have Salmonella enterica, which also causes food poisoning, and it has multiple long flagella. One of the reasons, if you ever known someone that's had Salmonella enterica, it's a really bad form of food poisoning. People eating eggs, things like that, oftentimes, undercooked eggs. I know that's one of the major culprits there. So anyway, there's some examples of flagella. So you can have single short, you can have multiple long, a lot of different variations in how they are arranged in these bacterial species. Okay, let's move on to slide 35. Let's talk about pili. Pili can actually twitch or cause a gliding move across surfaces. So here are some pictures of pili. They look like small cytoplasmic extensions that kind of cut off the cell here, kind of makes the bacterial cell look like a porcupine. Again, they can twitch, resulting in small movements or even gliding through mucilage. They are, again, very, very thin, and they allow the bacteria to stick to smooth surfaces. This is never mentioned here in this slide. They should have talked about this. This is a big thing they can do. Pili aren't used for movement. They allow these bacterial species to stick to smooth surfaces such as your teeth. So how do you even stick something as smooth as your teeth? Well, they have these pili. They also stick to the lining of the reproductive system in some cases. And so we also think that pili, and we know that they do, play roles in bacterial reproduction and disease processes. For example, how could pilus be involved in reproduction? The pilus operates like a penis, and that the pilus can extend outward and be like a tube and deliver DNA from one cell to the next. And that actually plays a role in evolution as well, like horizontal gene transfer. So there's the sex pili, or a sex pilus. It's just like these pili here, and they deliver DNA from one cell to the next. In disease processes, we have pili that actually inject toxins into host cells, causing disease symptoms, causing you to get sick. So that's how bacteria sometimes attack your cells. They actually shoot them full of toxins or DNA from the pili of these bacterial cells. So yeah, pretty brutal. So pili can be used to cause disease. Let's go to slide 36. Let's talk a little bit about bacterial reproduction. Well, when it comes to bacterial reproduction, it's not very exciting. Because guys, we don't really have sex or sexual reproduction. We don't have gametes. We don't have sperm and egg. Most often, the reproductive process of bacteria involves a process called binary fission, which is simply the splitting into two. The splitting into two is made possible by a really neat little tubulin protein called FTSZ. And FTSZ just simply squeezes the dividing cells into two progeny cells. Now, the binary fission process, the idea is that you start off with a cell, and then, of course, it's split two. What happens to those two? They four, so on and so forth. Well, if that's the main way that bacteria reproduce, this then can be used as a method for detecting and counting bacteria in samples. So let's say you did a water sample. You're testing the Morganton municipal water supply. Well, what you would do is you would measure an exact volume of the water sample into plastic dishes of agar, and then come back a few days later after they've been cultured, very much like what you all are doing in lab right now, and you'll see colonies. Well, you can assume that each colony with a petri dish, with these colonies, you can assume that each colony came from what? Yeah, each came from one individual cell. If we believe binary fission is the main way that bacteria reproduce, and that one bacterium can result in a whole colony, we assume that each of these colonies represents one original bacterial cell that was there. And so in the particular drawing that I gave you guys, I know it's not the best, but how many original bacterial cells were in that sample? Let's count it up. 1, 2, 3, 4, 5, 6, 7. 7 colonies. Therefore, there were probably 7 bacterial cells from that water sample. And depending on the volume of water that you put in the petri dish, you could then say that water is safe or harmful. I mean, it's not like just because you find harmful bacteria in water that you automatically blow the whistle and sound the alarm. It's the concentration of the harmful bacteria that really cause disease. Like there are times you'll consume a bacterial species like Salmonella enterica, but you'll never get sick because it's not in high enough concentrations to cause you to get sick, unless your immune system is severely compromised. But you can also use a fluorescent dye and fluorescence microscopy that this dye actually binds directly to bacterial DNA, and then you can literally just count them. You can actually look through the fluorescence microscope, count the glowing bacteria, just simply counting them up, and that's how simple that can be. So that's another technology, a more recent technology that we can use as well. Okay, so let's go into slide 37. Here are some examples of what we're talking about. So this is, it looks like E. coli or some type of bacillus rod-shaped bacteria undergoing binary fission. Could be happening right now. And you can see the colonies in this particular petri dish. We've got what? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, something like that, 14, 15. So that's probably a safe water sample. Over here, wow, that water sample led to lots of colonies. So very high concentration of bacteria here. This is probably unsafe if these colonies were an unsafe bacterial species. Or, like I said, you can just do the direct count through fluorescent binding dyes and fluorescence microscopy, and you can see individual cells. The dye binds to the DNA, which, of course, all these bacteria have DNA, so it works very effectively. So that's how we count or get an idea of the census out there of bacteria in a certain sample. Now let's look at just a couple ways that bacteria survive harsh conditions. Well, you know, in the life of a bacterium, things are going to get rough. The little puddle that you're living in is going to dry up, or it could be that the pH drops too low or the temperature gets cold. Lots of different things will cause bacteria to become stressed, and when they do, they have ways to handle harsh conditions. For example, they have aconites. Aconites are actual cells. They are considered to be true cells, and they have a very thick wall, a nice resistant thick wall. They're typically found among Lactobacillus bacteria, especially cyanobacteria, Steigonema, organisms like that, you know, photosynthetic photoautotrophs living in ponds. And when it gets cold, winter approaches, fall time's coming in, they'll begin to form these cells, these aconites, that are filled with nutrition. So they usually have lots of food. They're very large food-filled cells, and again have nice thick walls so they can remain dormant more or less throughout the winter. And then when spring comes, new filaments will come right out of the dormant aconites, and then it'll become an active cyanobacterial cell. You ever notice, like in the winter, like all of these algae die off and all the pond scum goes away, but then it seems like it just pops right back in the spring? Well, that's because the aconites are just sitting around dormant, ready for the warmer weather. As soon as it comes, they start producing new filaments, and the rest is history, right? They turn everything green again. But we also have endospores. Endospores aren't typically considered true cells, really. These are just simply very thick-walled structures that form within the cell. So you have and the endospore within the cell. I've heard some people call the endospores actual cells, but these are form within the cell. And these endospores within these cells have that protein coat. They can have a very long dormant span. Like with aconites, you're looking at maybe 6 months of dormancy, something like that or less. With endospores, you're talking about years, centuries even. They actually had endospores from thousands of years ago germinate and become living bacterial cells. So very long dormant span, very thick walls on these. You find them in some gram-positive bacteria such as Bacillus anthracis, Clostridium botulinum, and Clostridium tetani. Bacillus anthracis causes anthrax. Ooh, remember how scary anthrax was a few years ago? Well, you can collect the spores that are produced, the endospores from Bacillus anthracis, and that creates a powder, and when it's pure, it's like a white powder, and you can put that white powder in like a bomb, or you could put that white powder in even an envelope, and someone opens up the envelope, they breathe that in, they can get sick. That's what people try to do years ago with, I think it was President Bush at the time. People were trying to kill him that way, but of course they caught that. So anthracis is very bad when it goes through an open wound or when it's breathed in. If it's just through a small opening in the skin, it just creates a small skin infection. But if it gets into the bloodstream or you breathe it in, it's very deadly. Anthrax is very bad. It's used in bioterrorism. Clostridium botulinum causes botulism, which is a very harmful form of food poisoning. Clostridium botulinum hates oxygen, and so when you have a canned food that has a species in it, well, there's no oxygen in the can, and the Clostridium botulinum will start to eat the food. It produces ammonia and carbon dioxide, causes the can to bulge. So if you have a canned good that's bulging underneath some gas pressure, very good chance it could be Clostridium botulinum in there, so you do not want to eat that. throw it out immediately and tell the grocery store what happened, or call the company that that product came from. So you can cook it really high temperatures, and that will kill off a lot of the endospores, but the toxins are still left behind. So it's a very, very destructive form of food poisoning. If you were to consume Clostridium botulinum and the toxins, it actually causes paralysis of the respiratory and muscular system. So you can't breathe. You basically suffocate. It's just really, really harsh. And actually, Botox comes from Clostridium botulinum. You know, they actually use the toxin that could kill you, but they inject it into your face, and that paralyzes nerves in your face to remove the appearance of wrinkles. So Botox is actually a toxin, guys, that comes from this species. And Clostridium tetani causes tetanus, and of course you know the tetanus shot, how important that is. It's a nerve toxin that causes lockjaw, and it's where we have bacterial cells or even endospores. They enter wounds, especially deep puncture wounds like the rusty nail. So that's why it's important to get your tetanus shot on a regular basis. If not, tetanus would be a horrible thing to die from. Locked jaw, then eventually all of your muscles become locked, so it's extremely uncomfortable, extremely rough. It'd be one of the worst ways to die. So that tetanus shot is really a good thing when you think about the alternative. This is showing you cyanobacterial cells with spec cells like the heterocysts, which perform nitrogen fixation, and then aches and endospores. So I'm sorry, this is an aconite right here, guys. So here's the aconite right here. This has to perform nitrogen fixation, and this. Well, guys, we are going to wrap it up here. We won't quite get to, looks like we're going to basically be knocking on the door of slide 40 here. We won't quite get to that, so we'll just call it a lesson right here. If you have any questions for me, email me at mspath@wpcc.edu or bring your questions to class. We'll end with our little bacterial flagellum song. Everything is going out. Everything is going out. Plan, yeah. Everything moves along. Nice catchy song. All right, guys. See you later.